Welcome everyone, and thank you for joining us for today's webinar titled Measuring Energy Balance in Mice from VO2, VCO2, Food Intake, and Activity Data. My name is Martin Hess from Inside Scientific, and I will be your host for today's event. We're going to start just a minute or two early just to take care of some housekeeping details. We have a very busy agenda for today. We are happy to share that over 200 scientists from around the world have registered to participate in today's webinar, and I'd like to extend a warm welcome to those of you who are online with us. Thank you very much. Our session today is sponsored by Columbus Instruments, and we'll touch on the essentials of energy balance, including a review of the basic metabolic equations and an overview of various technologies available, as well as an overview of how to measure and interpret energy expenditure and metabolism in mice. We have two speakers participating in our webinar today. Our first speaker is Chris Adams, who is the Sales Manager, Marketing Director, and Production Coordinator at Columbus Instruments in Columbus, Ohio. Chris has 14 years of experience helping customers configure solutions for a variety of research applications that require robust and reliable equipment for measuring energy expenditure and metabolic function in rodents. Next, we will be joined by Dr. Marta Fiorotto, who is an Associate Professor of Pediatrics Nutrition and Associate Professor of Molecular Physiology and Biophysics at Baylor College of Medicine in Houston, Texas. Marta is also Director of the Mouse Metabolic Research Unit at the Children's Nutrition Research Center at Baylor College of Medicine. Dr. Fiorotto obtained her PhD from the University of Cambridge in the UK and completed a postdoctoral fellowship at Baylor College of Medicine, where she continues to work today. Dr. Fiorotto's professional interest is in developmental determinants of skeletal muscle growth, and the major objective of her research is to identify the mechanisms that underlie the various effects of nutrient intake on the growth and function of skeletal muscle.
Thank you, Martin, and welcome everyone. I want to thank you for your time today. My talk today will be centered around the CLAM system that we offer, and CLAMS, of course, stands for Comprehensive Lab Animal Monitoring System. The history of the system, as it first began as just a calorimeter called Oxymax back in 1987. It was the first commercially available system of its kind back then. And then about 10 years later, we collaborated with the Jackson Laboratory to make the first system capable of simultaneous VO2, VCO2, RER measurements along with food intake and activity. And then many, many years later, we've made a lot of advancements on that. And today we'll be covering just some of the most popular options, especially those relevant to Marta's research and the configuration of her system, and that's indirect calorimetry, food intake, food-controlled access, activity, and then finally data analysis. I'll begin with Graham Lusk's paper on the RQs resulting from the oxidation of fat and carbohydrates. And through his research, we ended up with this very handy table, which shows respiratory quotient down on the left column and how that equates to a percentage of fat and carbs that are metabolized. And on the far right, we've got heat per liters of oxygen according to those RQs. A quick concept of indirect calorimetry and how this works. Essentially, we have the subject in an airtight environment. It's ventilated by a precisely controlled flow of air. And then we measure the difference of oxygen and CO2 into the cage versus out. And here are the equations that are used for those. We have VO2, which is, of course, volume of oxygen in minus oxygen out. VCO2, which is the CO2 out minus the CO2 in. And RER, of course, is VCO2 over VO2. Of course, it can't be as simple as that, since there is an exchange of oxygen for CO2 during the process. The volume into the cage is not equal to the volume out. And because this exchange also displaces the nitrogen inside the cage, even though none is consumed or produced during respiration, it is displaced and it has to be accounted for. And that's where we come up with the Haldane transform there at the bottom. That's the volume out of the cage is equal to the volume in times the ratio of nitrogen in and out of the cage. We'll do a quick example here. I'm going to fly through a lot of math just for time considerations, but I'll show all my work, so if anyone wants to come back through the slides and check it out in more detail, they can do so. We're going to start with rather typical oxygen and CO2 in figures. and pretty ordinary oxygen and CO2 out. And in this example, we'll have the flow set at 0.5 liter a minute. First is the Haldane. We have to solve for the volume out so that we can do our VO2, VCO2 measurements. We plug those into the equation, and we find out that the flow out of the cage is just slightly lower at 0.499 liters per minute. So with that known value, we can plug those into the equations for VO2 and VCO2, and you get these following results for those measurements. And from there, it's a snap to get your RER. Just put CO2 over O2, and there's our RER. Normalization is a hot topic. There's a lot of discussion out there as far as what animal mass to use for normalization. For this example, we're just going to stick to the basics of just whole body mass. There's some people that use lean body mass, but you'll need a body composition analyzer to use those figures. But here in this example, we've converted the VO2 into milliliters per hour, divided it by the mass of the animal, and ended up with our normalized VO2. We'll go back to the Lusk table here, and it's important to note that a common mistake is equating VO2 to heat, and that's not true. Energy expenditure is heat. So in regards to the Lusk table here, we've got respiratory exchange ratio on the left, and I want to draw your attention to kcals per liter of oxygen on the far right column. We put those in a graph. It's incredibly linear, which allows us to yield an equation where we can plug RER in for X and solve for heat per liter of oxygen. Going back to our previous example, we had an RER of 0.8999. We plug that into our equation to get a calorific value of 4.9237 kcals per liter of oxygen. Now we can get heat. Heat is the calorific value times the VO2. And in this particular example, we have 0.63132 kcals per hour. And it's also common to go yet another step further to normalize that heat, where you have kcals per kilogram per hour. The most common way of calculating heat is using the Weir equation. And Dr. Weir's work was specifically to account for the oxidation of proteins, where Lusk was strictly fat and carbohydrate. And we have this equation as what we call the user-defined equation in our software. And it has the following equation where heat is equal to calorific value 1 times VO2 plus the calorific value 2 times VCO2. We'll plug some numbers in here, and first I'm going to start with the Lusk numbers that we got from the previous graph. We'll plug those into the Weir equation, and as you see, we get a heat that is remarkably similar to the one that we got from the Lusk table. However, a lot of people change their Weir constants, and these vary a bit depending on diet, animal strain, but they're typically in the neighborhood of 3.9 to 1.1. So we plug those into the same equation, and we get our heat figure, and we can compare the two to see how much of an impact those constants can have on your calculation of heat. Now we're going to look at a block diagram of how the calorimeter works. We've got the math down, so we'll see how this works in real life. And what we have here is an air pump, which is pushing air through a mass flow controller that then splits the air flow into 8 cages in this example. Those cages are then hooked up to a valved manifold for sequential measurements, so we can go cage to cage. Sample air is drawn by a pump through a drying device to remove all the moisture, and ultimately to our O2 and CO2 sensors where we get our readings. The cages in this example are measured sequentially, and you hear the term cycle time used a lot when you're looking at indirect calorimetry, and the cycle time is essentially the reference air, that's the measurement of air into the cage, and then the cycle through cage 1, cage 2, et cetera, until you get to the last one, and that's what's known as the cycle time. There are 3 different technologies out there for measuring oxygen, and they all have their own advantages and their own applications, and we'll dive into how they work and their pros and cons. The paramagnetic sensor is probably the most common one out there, mostly because it's maintenance free and it has reasonably fast performance. The The electrochemical cell is also very popular. It's best suited for small economical systems, so if you're on a budget, this is your sensor. And lastly, we have the zirconia high-speed sensor. This has the fastest scan rate of all three, and it's ideal for a large number of subjects. So beginning with the paramagnetic sensor, the way this works is we have a dumbbell suspended in a magnetic field, and it twists as it's exposed to oxygen. The more oxygen, the more it twists. We bounce a light source off a mirror that's attached to this dumbbell, and we monitor how much that dumbbell twists, and then by passing an electric current through that dumbbell, we can counter twist it back to center, and the amount of voltage necessary to twist that dumbbell back to center is proportional to the concentration of oxygen in the sample. The pros of this sensor is that it's maintenance free. There's no consumable element to replace ever, and it also has a user programmable range anywhere between 0 to 100%, although the typical range for most users would be between 19 and 21%. And in respect to our clam system, you get a scan rate of about 90 seconds per chamber. Not a lot of cons with this sensor. It's pretty reliable. The only drawback is maybe it's not the fastest. It's not as fast as zirconia, but it's still pretty fast. The electrochemical cell is your budget oxygen sensor. Essentially, it's a battery. Sample air diffuses through a membrane that drives a chemical reaction that produces a voltage, and that voltage is proportional to the oxygen concentration. Pros of this system that it's high accuracy at a low cost. The drawbacks are that like a battery, it does wear out. It requires periodic replacement, and it's a bit on the slow side. In our Oxymax system, it typically runs at about 3 minutes per cage. Lastly, we have the zirconia oxygen sensor. The zirconium dioxide conducts electricity at very high temperatures. Oxygen ions pass through it and creates a voltage. And essentially what we do is we're measuring two oxygen sources at the same time. We've got a reference gas and then the measured gas, and we're looking at the voltage difference because of the concentration difference, and of course that difference voltage is equal to the concentration of oxygen. The advantages to this sensor is that it's very, very fast. It can measure a scan rate of about 50 seconds per chamber. The drawbacks are that it's expensive to buy and that it does have a consumable element, and while it does last several years, it is expensive to replace when it does finally go. CO2 sensors are pretty standard across the board. CO2 will absorb infrared light at a very specific wavelength, and essentially all we do is shine that IR light, a precisely known amount, through the sample gas and measure how much is absorbed by the gas, and that will give you the concentration of CO2. And as I mentioned in the block diagram, we dry all of the gas before it goes into the sensors. We find that it's more reliable that way. We don't like the moisture passing through the sensors where they might condense. And it's also difficult to account for mathematically because it does vary so much, and the sensor technology isn't outstanding. So what we use is the Nafion tube dryer. Essentially, it's a very special plastic that will selectively remove water vapor if you can create a moisture differential between the inside of the tube and the outside of the tube. So we place an outer tube in which we pump dry purge gas that's run through columns of dry right to create our moisture differential, which ultimately dries the sample gas. And here's an example of some of the data that comes out of our Oxymax clams calorimeter. You can see basically the VO2, O2 in, out, deltas, the accumulated, and if you pan all the way to the right there, you'll see RERs, heat, and the flows measured in real time. It's worth noting that these are real-time readings. They're shown as the experiment's going on, and all of this can be exported as a CSV file at the end. We also validate all of our calorimeters before they ship out by CO2 infusion. And essentially what we do is we have a machine we've built here that uses very precise injections of 20% CO2 to simulate the exhaled gas from a mouse, and that 80% nitrogen also displaces oxygen, which allows us to simulate the O2 consumption by an animal. And this is a shot of the spreadsheet we use in-house, and essentially there at the top we have our known values. That's the gas that we're pumping into the cage. The next section down is our targets. These are theoretical yields that we should get with the values that we've used before. And then the last section there is measured And essentially what we do here is we just run a test, like there was an animal in the cage. We run it for a few hours and see what measurements we get. And we need them to be as close as possible to theoretical. And in this case, you can see we ended up with 0.9427 RAR versus what we expected to see at 9.43, which is remarkably close. Next, I'll talk about feeders. There's a few different feeders out there on the market, and we offer all the solutions that are out there. There's overhead feeders, and there's also our center feeder cage, which we'll talk about here in a moment. But I'll start with the overhead feeder. And there's pros and cons to each one of these. The overhead feeder, one of the pros is that it keeps the food in a familiar location, and it allows you to use a standard shoebox cage and the use of bedding, which can lower the stress. It allows you to use pelleted food, and the maintenance is very, very easy. You can just throw these cages into a cage washer. Cons, however, is that it is vulnerable to foraging. The animals can chew down a pellet to a smaller size chunk and then pull it out with their hands, and they drop it into bedding. This is worth noting because this can cause some problems. Not only is that food removal recorded as food being eaten, but if you've got food access and you're trying to fast the animals, if they stumble upon that chunk of food later, they'll eat it. And you'll see that metabolic spike happen in the calorimetry data, and it'll be difficult to account for. You'll know what it is, but it does make it difficult to adjust for later on. Also, the overhead feeder has a limited amount of spillage collection, and it can overflow. And also, because it is suspended, it can pendulum a bit, and it takes them longer to settle. Conversely, our center feeder cage was designed to combat all of those problems, but again, it also has its pros and its cons. The use of powdered or crushed food makes foraging a minimum problem, and there's a very large spillage reservoir to account for all of the spillage. And plus, since it's on a balance and it's beneath the cage, the weight stabilizes much, much faster, which gives you a higher resolution for feeding bout data. Cons, of course, is that the food is at the floor, it's in an unfamiliar place, and it takes the animals some time to get acclimated to the feeder. Also, the food must be powdered or crushed. You can order powdered food, or you can take some pellets and either run them through a coffee grinder or smash them with a hammer in a plastic bag. There's also no bedding, so it takes some acclimation to the floor, and the acclimation time has to be accounted for. You've got to set aside time to get the animals acclimated to that environment. Our food access control allows you to cut off access to the food either by way of a shield that raises in the center feeder or by twisting the suspended feeder with a servo into a shield. And you can make those events happen based on time, based on mass, or both. Additionally, we have a section where you can enter in an energy coefficient for the food that you're using. So if you wanted to do a caloric restriction, you can do that by entering the energy coefficient. And we also have yoked and paired feeding, where you can choose one of the mice to dictate when the other mice have access to their food based on the master's feeding pattern. Here's a look at some of the feeding data that we give you. We actually give you 3 different ways of collecting the feeding data. This is the synchronous data. This is what's reported with the calorimetry data. So you can see how much food was eaten during that interval and the accumulated amount of food eaten thus far. We also have the asynchronous feeding event log, which is time-stamped events of feeding bouts. You can see the amount, duration, and how much has been eaten so far. And then lastly, we have a periodic user-defined interval. And this is one where you can go in. If neither one of these 2 options is quite what you're looking for, you can make your own interval length, and it'll collect feeding data at that interval and save it in its own separate file. Next, we'll talk about activity monitoring. And activity monitoring in the CLAN system is done by infrared photocells. And we can configure the activity in a variety of ways, but typically X, Z axis are the most popular, and that would be the X axis for horizontal motion along the long side of the cage. Z axis would be rearing. We've also got Y axis, which of course is the short side of the cage. The Y axis can be very useful if sleeping bout information is of the highest importance to your research. Otherwise, for just gross locomotor activity, since it is the short side of the cage, it doesn't offer a lot. There are some cost savings to be had by getting rid of that sensor. And like the feeding, we have several different activity data sets that we give you with the system. There's data that's synchronized with the calorimetry data, so you can see here in this data set the XTOT, that's the total beam breaks that happened during the experiment. And it's worth noting that the total beam breaks is the combination of ambulatory beam breaks from locomotor activity plus any of the fine repetitive stereotypic movements like feeding or grooming. Next we have the XAMB column, that is strictly ambulatory motion from locomotor movement around the cage. We also report that for the y-axis if equipped. Then we have the z-axis, which is rearing events. And then finally, if the system's equipped with a running wheel, we'll give you the revolutions per interval. We also have an inactivity data file where we record sleeping bouts. And this method was developed by Alan Pack at University of Pennsylvania. And he used the CLAM system to develop this screening method while he simultaneously measured EEG readings to validate these sleeping bouts. And he came up with a remarkably high correlation between inactivity bouts and sleeping. And here's a screenshot of our software here, where you can see the sleeping bouts that are time-stamped there in the large window on the left. And if you pan to the right there, you'll see these sleep threshold epochs. And this effectively allows you to set the amount of inactivity that determines if a sleeping bout has occurred or not. And then down at the bottom, we keep track of our light and dark cycles as well. The activity data in the high resolution file, in this particular example, I've set the frequency at 10 seconds. And similar to the terminology used on the synchronous file, we have X total, X ambulatory, and rearing events. And also the running wheel is also used for spontaneous locomotor activity. It's very useful that it provides an infinite floor space, which both allows the animal to ambulate more and also reduces stress. We report the data synchronized with the calorimetry, of course, but there's also a high-resolution activity data formatted similarly to the high-resolution activity data that I showed just previously, and Marta will touch on the use of that data file in her presentation. Lastly, we have the CLACS data review and stat program, and this is a separate program that comes along with our CLAM software. It opens the data files natively and allows you to do quick screening for compounds. So if you just want to see how the experiment went without having to open up Excel, crunching in all the numbers and the figures, you can just open the data files in this program and get a quick and dirty analysis and see what happened. Additionally, if you have collaborators with the same system, you can compare multiple data sets and share them back and forth. You can also open multiple data sets and compare them side by side. And lastly, we have some stat analysis tools built in for you, so you can do some quick and dirty stats. Also, you can generate all of your graphs necessary for your research papers. Now, all the features I've spoken about here are equipped on Marta's system, and she'll discuss how she uses these tools to measure.
And thank you to all of you for participating in this webinar today. My presentation today is going to be divided into three main parts. First, I'm going to go over some basics of energy metabolism and how to assess it. And I'm then going to review examples of data that we've generated with our system over the years with the goal of differentiating between those measurements that are truly quantitative from those that are qualitative, but that really provide very important information that will help you identify the mechanisms responsible for the changes in energy balance in your mouse model of interest. And finally, I'm going to share some tips, best practices and suggestions for getting biologically relevant data, and this is sort of based on our experience with the system. I'm going to begin by reviewing a few basics of energy metabolism and how it can be assessed. First, however, I would like to emphasize that when you're evaluating energy balance data, you should always keep in the back of your mind that nature does not defy the laws of thermodynamics. And the law that's most important as far as energy balance is concerned is the first law of thermodynamics, which states that energy can be neither created nor destroyed. But it can change forms, and it does flow from one place to another. So the total energy in an isolated system always remains the same. So basically remember that things have to add up at the end of the day. So what does this mean for an organism? It essentially means that all the metabolizable energy that enters into the body is either expended and ultimately converted to heat, or it can be stored in the body tissues. In the adult, this excess energy is largely stored as fat in the adipose tissue, although you've got to remember that with all adipose tissue, there's an obligatory component of lean that's also accumulated. And there's also a small component of energy saved as glycogen. These changes are then manifested as a change in body weight and composition. You've got to remember that in a growing individual, this non-expended component is important because it represents physiological growth. So, in a body in which total intake is equal to expenditure, there is energy balance, and so body weight and composition remain constant. However, if you consume energy, if they're consumed in excess of the energy expended, or energy expenditure decreases without a compensation in intake, there is a positive energy balance. And then this excess energy is converted to fat, and where it expands, the adipose tissue depots and is manifested as weight gain. When energy balance is negative, because energy intake decreases relative to energy expenditure, the extra energy required to meet the body's needs is then met by mobilizing the body's reserves, and it results in weight loss. So you've heard from Chris how the clams can provide you with some of the information needed to determine how to measure energy balance. However, there are additional parameters that you must have just as accurate knowledge of in order to estimate and evaluate your energy balance data. And so let's look at what these are. So let's begin with the metabolizable energy intake. The clams gives you a measure of food removed from the feeder, which with the center feeders we can reasonably assume represents food intake. However, to estimate the amount of dietary energy that's available to the body, you will need to know the metabolizable energy density of the diet. And then with these two parameters together, you can calculate intake. So what is metabolizable energy? This is the energy in the diet that is available for heat production, i.e. energy expenditure and body gains. And the most common approach to derive metabolizable energy intake is to calculate it from the macronutrient composition and the Atwater factors shown here. And these Atwater factors represent the heat of combustion for protein, fat, and various carbohydrates and alcohol, corrected for the losses due to incomplete digestion and absorption, and the urinary excretion of urea, which results from the incomplete oxidation of amino acids. The values shown here are just the traditional ones used, although I recognize there's a long-standing debate over what's most appropriate. Ideally, I would say, though, if you want a true, accurate estimate of energy intake, you should have the composition of your diet analyzed. As Chris has described, the clams provides you a measure of VO2, VCO2, RER, and heat. However, it's also very important to consider if protein oxidation could be contributing to energy production in your particular model, because the equations used assume this to be negligible. In most circumstances, it's okay to make this assumption, but this may not always be the case, especially if you have a lot of weight loss. The other outcome that we often want to know is energy balance, i.e. the amount of energy deposited or mobilized. Now, the clams gives us energy intake and expenditure, and so you should be able to calculate the balance. However, the values derived this way are usually quite imprecise because they include the sum of all the errors and assumptions in the measurements of intake and expenditure. So ideally then what you want to do is you should accompany these measurements with body weight and fat and lean mass, and these should reflect the changes in balance as would be predicted by the first law of thermodynamics. Now, when evaluating your data, it's important to remember that differences in energy expenditure can result simply due to differences in the metabolic mass of the organism. So to determine if there are differences in energy expenditure that cannot be accounted for simply because of differences in metabolic mass, which is really the question we're usually after, you have to normalize for metabolic mass. The data from the clams, as you saw, gives you either total or adjusted for body weight. Ideally, however, you should get measures of fat and lean, and these should be used for normalization by analysis of covariance. There are a number of excellent publications, such as the one shown on the screen here, that go into the reasons why this is a preferable approach, and they also give examples of misleading conclusion you can arrive at if you don't. So one of the goals of performing these measurements is not only to get total energy expenditure, but to then identify what's responsible for any of the differences you may find. So at this point, we'll look at the various components of energy expenditure, and these include the basal metabolic rate, which is the energy needed to sustain metabolic activities and to maintain various physiological function. And it's usually the largest, it's always the largest components of energy expenditure, on the order of 60 to 70%. There is thermogenesis, which is the energy expended to maintain body temperature. The thermic effect of food, also called dietary-induced thermogenesis, or the specific dynamic action of food. And this is the energy expenditure associated with the digestion and assimilation of food. And it usually represents about 10% of energy expenditure. And then finally, there's the most variable component, which is the energy expended during physical activity. So how can we get a measure of these various components? So BMR is going to reflect the energy expenditure of an organism that is at rest, which is no activity. It's in the postprandial state, so there's no thermic effect of food, and at thermoneutrality. However, as shown in this graph right here, the thermoneutral zone for a mouse where there is no thermogenesis occurs between about 28 and 34 degrees centigrade. In reality, however, most mouse rooms are not at thermoneutrality. Moreover, there's usually no bedding in the cages, and as mice are individually housed, they can't huddle. So there's no microenvironment to reduce heat loss. And thus, even though the temperature inside the cage can be a couple of degrees warmer than room temperature, it's usually below thermoneutrality. So thermogenesis almost always inevitably will be contributing to energy expenditure, even measuring basal metabolism. And so we call this resting energy expenditure or resting metabolic rate. And again, interpretation requires normalization for lean and fat mass. In this next slide is a protocol that we use to get estimates of RMR using a CLAMS configuration that's optimized for longer-term daily energy expenditure measurements. We first acclimate the mice to the CLAMS cages so that their activity and stress levels are back to normal. We close the feeders at lights on when the mouse is entering its resting phase and activity is lower. We continue to monitor energy expenditure then activity for 7 to 9 hours. And we then take the average of the lowest two values over the last 3 to 4 hours. And we've determined that in most cases this coincides with a period when there is no activity. Again, the mice shouldn't be disturbed when you're trying to do these measurements. I should note that to get very accurate measurements of RMR, a modified instrument configuration optimized for rapid short-term measurements, which is not really suitable for chronic measurements, might be preferable, and this is discussed in this article here. Determining how much of total daily energy expenditure is due to physical activity is not straightforward with a multi-cage system because of the lag time between when an activity occurs and when gases are measured. Additionally, activities vary in terms of their energy cost and their duration. A continuous measuring system is better suited for this purpose, although, of course, it has its own set of limitations. And again, I would refer you to these two articles for greater discussion. Nonetheless, as you've heard, cages can be equipped with systems in which cage activity or wheel running is monitored, and comparisons between control and treatment groups can then provide you information on whether there are differences in activity levels that could be contributing to the variability in your total energy expenditure. And I'll give you some examples. However, again, if you're interested in the energy cost of locomotion, you can determine this with a metabolic treadmill in which you set the activity level at whatever you want, and then you measure VO2, VCO2, and you sample measure these at very high frequency. the thermic response to feeding can be estimated from the changes in energy expenditure following the consumption of a standard meal after a fast, minus resting metabolic rate and activity energy expenditure. Because rodents requires a concurrent estimation of RMR and activity, it's a parameter that's not easily determined with an instrument configuration optimized for chronic energy expenditure measurements. However, a system in which frequent sampling and rapid measurements are possible, you can determine this, and this again is discussed in these two articles here. So in conclusion, an accurate assessment of energy balance and its components is technically complex. While the system is straightforward to use, correct experimental design and an initial preparation is equally, if not more important, and really merits careful consideration. But when done correctly, you can get intake and energy expenditure measurements with sufficient accuracy to provide much insight into the energetic consequences of your particular genetic manipulation, diet, drug, or treatment of interest. So now we've determined what we need. So let's just look at some examples of data that we've collected in various studies and how they've contributed to understanding the effects of various interventions on energy balance and understanding the mechanistic basis for these differences. So here is an example of a, a genetic mouse model in which the receptor for the orexigenic hormone ghrelin has been ablated. All the mice that aged, as they aged, they gained less weight, and this difference in weight gain was largely due to accretion of less fat. So in theory, you might expect these mice to eat less. However, when food intake was measured, it was not different between the controls and the ghrelin receptor null mice. So this basically suggests that it's not differences in food intake, but therefore possibly due to differences in energy expenditure. So when total energy expenditure was measured, indeed, it was higher in the null mice during both the resting phase and the active phase. And then the measurements of RMR actually showed that the ghrelin null mice have a higher resting metabolic rate than the controls. And there was no differences in activity that could have contributed to this difference in energy balance. Now, in addition to measuring total food intake of an ad lib fed animal, if you get high frequency food intake data, there is much additional information that describes feeding behavior that you can get. And some of the parameters that can be extracted include the number of feeding bouts or meals per day, the amount of food eaten per meal, as well as how much they eat in a day, the duration of the feeding bouts, the length of time between feeding bouts, and then you can always get the diurnal eating patterns. And here is an example of a study in which feeding behavior was assessed in the same GHSR null mice mentioned previously. And at the start of the experiment, we defined a feeding bout as an intake of at least 20 milligrams, with the balance being stable for at least 10 seconds. And what did we find? As you saw previously, there was no differences in total food intake. However, the average interval between meals was longer in the ghrelin receptor null mice, and as you would expect, they had fewer meals per day. However, each meal lasted longer, and they ate much more at each meal. And so these data provided them the information, the role of the GHSR in regulating feeding behavior, and which may also have contributed to the observed differences in energy homeostasis. Now, the continuous monitoring of food intake also allows one to evaluate the diurnal pattern of food consumption. And in this example, the food consumption pattern was monitored in a genetic mouse model in which the hepatic steroid receptor coactivator, SRC2, was deleted. And it was observed that the peak feeding in the SRC2 null mice was advanced by approximately 85 minutes. And these data mirrored the changes in wheel running patterns, and together with cellular and molecular data collected, demonstrated that the SRC2 is a critical positive regulator of the mammalian circadian clock. Now, as previously mentioned, evaluation of the contribution of differences in activity to energy expenditure is an important part of understanding changes in the energy economy of the organism. And although it may not be possible to ascertain the contribution of differences in physical activity quantitatively, a couple of examples will show you that I think very meaningful data can be derived by quantifying it. So in this first example, the effects of fetal growth retardation on postnatal growth and body composition was being investigated. And the female growth-retarded offspring had regained their normal weight by weaning, but as they became adults, they gained more weight and were heavier than the controls, and this difference was due to an increased accumulation of fat. Energy intake and expenditure were measured both at puberty and as adults. And first of all, there was no differences in food intake were found. However, energy expenditure in the fetal growth-restricted animals was significantly lower than in controls. Cage activity measurements then showed that the activity was actually lower in the growth-restricted mice, and the time when the differences were maximal coincided with the times when there were differences in energy expenditure, strongly suggesting that the activity differences contributed to changes in energy balance. And indeed, when these investigators did a regression analysis, it showed that the variation in physical activity significantly contributed to the variation in adiposity. On the other hand, you can have instances when you have to consider food intake, activity, and the various components of energy expenditure in order to explain what's happening with energy balance. An example of this is the juvenile growing mouse model for Duchenne muscular dystrophy. And in this instant, the young MDX mouse is significantly lighter than the age-matched control, despite the fact that they're eating the same relative amounts. And over 24 hours, they're only half as active and therefore should be expending less energy. However, we look at their total energy expenditure, we see that this was higher, and this was due largely to this increase in resting metabolic rate. And in this case, study was done so that we could estimate energy balance. And indeed, we did find the energy balance was substantially less in the MDX compared to the controls, and this difference likely contributed to the lower growth rates. Now, as you've heard, an additional option is the possibility of incorporating running wheels in the clams, and therefore being able to measure spontaneous cage activity that may reflect behavioral differences and voluntary physical activity, which is also influenced by exercise capacity. And an example of some data we recently collected is shown here, and in this study, the treatment resulted in approximately a 50% reduction in wheel running over a 3-week period. At the same time, we measured cage activity and determined that neither horizontal, ambular activity, nor rearing in the z-axis was affected by the treatment, although there was significantly more rearing in the wheel runners. Now, the wheel running was associated with significantly higher food intakes, as shown by the solid bars as opposed to the crosshatched, which is the sedentary animals. But the difference intake between the controls, the green, and the treated animal, the pink ones, was not as great as their differences in activity levels. Thus, you would expect that the treated mice would be in a greater positive energy balance, and this was indeed manifested by the differences in fat gain over the period by the treated mice compared to the controls. So we then asked ourselves if these mice didn't run because they weren't most motivated or because they couldn't. And we reasoned if it was lack of motivation, it would be manifested as a decrease in the number of bouts of running. So we set up the system to record activity at very high frequency. It's in 10-second bins. And we also defined a bout of activity as at least 4 revolutions in 10 seconds. And what we showed, much to our surprise, was that the number of bouts initiated per day was not different between the two groups, but the number of revolutions per bout was significantly less in the treated mice and could point to a differences in exercise capacity. However, as we had the high resolution data, we also evaluated if the amount of time that the mice were inactive was different. So as you heard from Chris, Pat determined that one could derive information on the amount of time spent inactive, resting, sleeping, whatever you want to call it, by monitoring the duration of bouts of complete inactivity. So using their suggested parameters and the high-frequency activity data, we determined that the treatment group spent more time sleeping. Moreover, the control mice that had the wheels slept less than the sedentary controls, but this difference was not seen in the treated group. Now, whether the increased sleeping contributed to the decreased running or vice versa, we can't tell right now. A final option that's very helpful, and you've heard about, is the possibility of controlling the access of the mice to their food. And the following examples of studies that we have performed. We've done pair feeding, in which the amount of food consumed by one genotype, one experimental group, was fed to a control mouse. And the response then will directly reflect whether a metabolic phenotype that you might be seeing results from differences in food intake or from differences in other components of energy expenditure. You can also do meal feeding, in which the daily allowance of a diet is given at specific times of day, over varying lengths of time, or a specific number of meals, and the effects on the varying components of energy expenditure determined. And then you can do restricted feeding experiments, in which you just give a fixed amount of food daily. And this is done either in absolute terms, per body weight, or in energetic terms. Using this automated approach has a number of advantages. First, it reduces your manpower needs, and reducing manpower, you also reduce mistakes. Secondly, it enables feeding times at times that are biologically relevant to the mouse rather than to the humans. With pair feeding, diurnal feeding patterns of both groups is maintained equivalent. And when there is a restricted feeding protocol, the food can be made available at multiple intervals rather than just as 1 or 2 meals a day as is when done manually. And this is less physiologically and metabolically stressful for the mice. There's also less interference with mouse cages, and this introduces fewer disruptions. So finally, I'm going to share with you some of the do's and don'ts in designing experiments that are important to achieve accurate, precise, and I think most importantly, biologically relevant and valid information. Now, what I'm going to share has been our experience for over about 6 years, and I want to say by no means is this the only way of doing things. And in fact, there might be better ways, and if you figured out better ways, I'd love to hear about them. So one of the most important things that we have found is that you acclimate the mice to the clams' cages before you start your measurements. The mice are faced with numerous changes when they go from their regular cages into the clams' cages, and these are listed here. And they can respond to these changes in a variety of ways, and it's critical, therefore, that when you come to doing your measurements, you're not measuring the animal's response to this stress. And so the protocol that we recommend is that 1 week, it's a 2-step protocol. In 1 week prior to the start, you singly house and feed the powdered diet in their home cage. Then 3 days before the start of your measurements, you place the animals in the clams cages, and then you collect data. After 3 days, you collect data for further 48 hours. And the next slide shows the sort of variability and changes that you can expect over 5 days in the clams for food intake, activity, and daily energy expenditure. Please note this is a best case scenario with mice that were very close in weight and of the same age. And they had been through the first step of acclimation. And as you can see, the way these 3 parameters respond over time is different. Mouse variability is high at the beginning. This is shown by the standard deviation bar, and I put coefficients of variation across the bottom. So the variability is high at the beginning, and also the absolute values are high at the beginning. But then they do even out. Now, it's important to also recognize that there can be significant animal-to-animal variability in behavior that can introduce significant variability and error, and ultimately can invalidate your results. And some of these are listed on the slide. Some mice never adapt, and they become depressed, sleep, and don't eat. Some mice love to dig in their food, and they'll empty out their hoppers very quickly, and then if you don't watch it, although that's not measured as intake, they'll be left without food. Some small mice, in the 10 to 12 gram range, depending on the type of floor you have, can actually find little holes, and they can sneak through those, and then they get stuck underneath, and then they can't eat and drink. Small mice sometimes can't reach the standard sipper tubes, they're water, and so you need to have especially long sipper for small mice. And some mice on restricted intakes get so desperate that they learn to defeat the food access control system. This does not happen very often. So what we actually recommend is that you monitor your mice very closely during the measurements, and you weigh them before and after the clams measurements, and look for abnormal changes in weight, as this is usually an indication that something didn't quite go right. Environmental variables also can influence your experiments. It's important that temperatures maintain constant so that the contribution of thermogenesis is constant. Inappropriate light exposure and noise can alter behavior, and these should be avoided as they can introduce spurious changes in activity and energy expenditure. Now, there are a number of considerations that's important to give thought to before you begin an experiment. First of all, when you have a multiplex system, the frequency of energy expenditure measurements on individual mice will depend on the total number of mice that you have. So it's important to have an idea of how frequently you need an observation, and therefore how many mice you can have in your system. It's important to decide if you need high-frequency measurements, for example, for feeding behavior or activity assessments before the fact. You can't go back after the fact and get the data. Thirdly, when doing pair feeding studies, it's important to ensure a priori that the intakes of your master mice, i.e. those that determine food intake for the pairing, are within the expected norm, and that one of your master mice isn't one of these diggers or a depressed mouse or whatever, but that really will mess up your pairings. Fourthly, in order to enable mice to have access to the feeder, but not to make it so easy for them that they sort of sit and play with it or sleep on it, there are these spacers, and you can alter the height of this access to the feeder, and this should be customized for the size of the mouse. It's important to use different size spacers according to the weight of the mouse, and with growing mice you may actually have to change the spacer a couple of times during the experiments. It's important to sieve your diet if you make it yourself to ensure it has no lumps. If there are particles that are large enough for a mouse to carry, they can remove them and drop them in the cage, and they'll go through here, and although they can't eat them, as far as the clams is concerned, the mouse did eat them. And then finally, some general comments that are not only applicable to the clams, but to conducting these sorts of metabolic experiments in general. First of all, the timing of the measurements relative to the appearance of the phenotype should be given very careful consideration. Measurements after the phenotype has occurred could reflect an adaptation to the change rather than the cause. And finally, as it's likely that you may need to repeat an experiment in order to have an adequate number of replicates, it's important that all your experimental groups are included in each batch. Animal diet, environmental, and instrument variability can introduce batch-to-batch variability, but you can adjust for this statistically provided you have representation in each batch. And so with this, let me thank you all for participating. I'll be happy to answer questions, but first I'm going to hand off to Martin, who's going to be the moderator for the question and answer.
Let's start with a question for you, Chris. Essentially, what is the configuration of a standard clams system?
I get probably asked that question every day. It's one of the most popular ones. These systems are all built on a semi-custom basis to include as many or as few features that are needed or necessary to perform the task at hand. Typically, it's the budget that runs out before your wishes and hopes. But in terms of just energy expenditure strictly, all you really need is the calorimetry system. That's VO2, VCO2, and RER, which you can plug into those equations that I showed earlier and get energy expenditure. But in terms of energy balance, I would think the very bare minimum that you need is food intake and animal activity. Running wheel would be a great idea for that, so you can get the spontaneous activity and get that VO2, VCO2, RER data. And then beyond that, you can also do the food access, which Marta has. That would be an additional option you might consider. But the bare option, the standard clams for energy balance, would be calorimetry, activity, and food intake.
Okay, perfect. And I suppose one could start with those options and then keep on adding on as need be if necessary.
Absolutely.
Okay. Marta, you mentioned adjusting for thermoneutrality in your presentation. How do you manage that with a system like clams? Can you start by elaborating?
So, so if your experiment calls for it, sort of depends. You can just adjust your thermostat if that's possible, or there are chambers that allow you to do that, and I think Chris can probably address that better than I can.
Yeah, raising the temperature in the room isn't always a feasible option, especially at night. Sometimes the HVAC is automated and the heat kicks off in the winter and the room gets exceptionally cold, or vice versa in the summer or tropical climates. It can get very warm, warmer than you want. We have these environmental enclosures that we build now, which places the cages inside of a temperature and light-controlled enclosure that allows you to reach those thermoneutral temperatures, irrespective of the room temperature. You can also do cold challenging, for example, in those same enclosures, so they're very versatile. And then the light and dark cycle can be programmed in the software as well, so if you're looking at those circadian rhythms, you can control the light and dark cycle. And I'll skip ahead to one of the other questions that's come in, break protocol a little bit, but there was also a mention of group housed animals and how breaking them out into singly housed animals affects food intake and body weight. And there is an impact, especially if you just jump right into putting them in the clam system. And this highlights the importance that Marta spoke about acclimating the animals to the cages. When you pull them out, there's, of course, the stressful impact from no longer being group housed with their buddies, but the inability to huddle really impacts their resting energy expenditure because they have to burn more calories to keep their body heat up. So this temperature-controlled enclosure does help combat that. You can raise the temperature of the environment to help keep them at a thermoneutral state.
Okay, perfect. There's another question here about temperature and thermoregulation, and it has to do with housing without bedding versus with bedding, and the ambient temperatures required, one versus the other. Can you comment on that? Is there a difference? Any experience there?
The bedding does allow them to nest and keep some of that their body temperature up. The absence of the bedding is more of a stress issue, I think, than a temperature issue, so long as you have that environmental enclosure and can crank the temperature up a couple of degrees to compensate for it.
Perfect. Okay.
Can I just jump in here a minute?
Absolutely.
And say that we've done some studies and in which we've used some of this new bedding, which is sort of like the shredded, long strings of shredded paper. And we have had a study that's used that, and sometimes the mice drag it into their feeders, but that doesn't happen very often. And sometimes it can mess up your activity, but we're actually evaluating it because it may be feasible to include some of this sort of shredded spaghetti bedding in these cages.
Perfect. Okay. Thanks for your answers. Marta, question for you. What weight range of mice can be measured with a system like clams? And then also, we had a question here from Peter, and he asks, at what age can you start sort of measuring with mice? Or can you start the experiment?
So age and size go together, and really size is the limitation rather than the age. You do have to alter your flow parameters in your calorimeter because they are smaller, but I will tell you that we have successfully measured animals, again using floors where they can't get underneath, as low as about 10 grams right at weaning. So we've taken them at weaning and we've put them in the clams cages and done measurements.
Okay.
It takes longer because they're then adapting to everything in the clams cages, but it's doable and you get very reasonable data. And big, Well, we've gone up, we've measured bob mice in there, and they're very round, and I think we can, basically, we've done 60 grammars in there. Yeah.
Okay.
So I would say 10 to 60 grams, but you do have to adjust your flow parameters so that they're sensitive to the sort of changes that you're looking for.
Okay, perfect.
In energy expenditure.
I'll jump in there too, and I'll up that maximum figure. I've had customers with mice in there as large as 80 grams. In fact, that center feeder was designed specifically for the OB mice. As they grew in size, they were too big to eat from an overhead feeder. The only way we could get them to eat is if we just put the food directly on the floor right underneath their mouths. And those animals got up to easily 80 grams, and the system worked wonderfully.
Okay, fantastic. I'm looking at the time. We might make this our last question, and I guess, Chris, mostly for you. Aside from the data that was presented today by Marta, which was really fabulous, what additional parameters can be monitored during such experiments?
Yeah, clams has come a long way since the initial systems for just food intake and activity. These days, besides the environmental enclosures, which I mentioned, we also have telemetry for the system, so you can get core body temperature and heart rate, especially useful if you are doing experiments at not thermoneutrality, like a cold challenge or something like that. Heart rate, of course, being also very useful for that spontaneous activity in mice. We also have multiple diets available for the cages, so if you're looking for a meal preference, you can combine that with the food access to sort of force them to eat one diet versus the other. We've also done some special modifications on a custom basis for customers that need a tether to go to the animal, whether they need to do some sort of a fusion mid-experiment. We can make custom lids to accommodate a tether. And also, as Marta mentioned briefly in her presentation, the use of treadmills. We make airtight modular treadmills that plug right into the clam system. In fact, you can even control the speed from the clam software, so everything is automated.
Fantastic. Okay. We do have lots of questions here, so just a reminder to everyone that we are going to send a Q&A report to all of you after the event. At this point in time, I think we'll wrap up our session. I want to thank everyone for taking time out of their day to attend this webinar. We hope you found the information provided today valuable. I would also like to take this opportunity to thank you, Chris, from Columbus Instruments, for your time today and for sponsoring this wonderful event. A huge thank you to Dr. Marta Fiorotto for her very informative presentations and for sharing some of the data that you've collected in your lab with the CLAM system. And again, just a reminder, today's webinar has been recorded and will be made available for viewing at InsideScientific.com. The slide presentations and answers to all questions submitted will also be made available, so please look out for an email from us with links to those resources. I would also ask that you please take a moment to complete a brief survey that will follow this webinar. We would like to obtain feedback from you so we can continue to put together meaningful events and programs for our scientific audience. So with that said, it's 10 minutes past the hour. Thank you very much. Have a wonderful day, and we look forward to seeing you again at another one of our Inside Scientific webinars. Thank you.